Hello, welcome. Today we are celebrating the plant cell focus issue on photosynthesis. This is the October 2024 issue of the plant cell. The editors of this focus issue are Ralph Bach, Roberta Croce, Nancy Eckert, J. Clark Lagarius, Sabia Merchant, and Kevin Redding. And as I said, it's online now. Today we have three speakers who will be talking to us about their work. Lin Zhang, David Rollo, Clara Hot, sorry, Craigo. But we'll start with an introduction to the focus issue by Roberta Croce. Our moderator today is Guy Levin, who I will introduce in just a moment. After the webinar, we will post this recording to YouTube. If you go to our YouTube channel, Planté, you'll find recordings of all of our focus issue webinars as well as other webinars. We want to make a special thank you to ASPB member attendees. If you're interested in joining ASPB, if you use the code PRESENTS10, you get 10% off your membership dues. If you need a certificate of attendance, an email will go out tomorrow with instructions for how to obtain that. If you have technical problems, try logging out and logging in again. I'll be monitoring my email as well. And now I'm going to turn it over to Guy. Guy is currently a postdoc at the Technion Institute of Technology in Israel, and at the end of this year, he'll be moving to Berkeley to do a postdoc with Chris Nayogi. So I'll stop sharing and turn it over to Guy.
Thank you, Mary. First we're gonna have a little introduction by Roberta Croce. Roberta is a reviewing editor for The Plant Cell. She's a professor of biophysics of photosynthesis at the Vrije University in Amsterdam. She studied chemistry at the University of Padova and completed her PhD in plant biology at the University of Milan. Her research focuses on the molecular mechanisms of photosynthesis, especially the light reactions, which she studies at different levels of complexity, from single molecules to whole organisms. Her research uses an integrated approach, including molecular biology, biochemistry, and advanced spectroscopy and theoretical methods. And Roberta, the floor is yours.
Welcome everyone to this webinar on photosynthesis. My name is Roberta Croce and I'm one of the editors of the focus issue on this topic. Photosynthesis, as you may remember from your school days, is the process that sustains virtually all life on our planet. However, despite its fundamental role, there are still many knowledge gaps in our understanding of the process. For example, in how photosynthesis interact with other metabolic pathways and how photosynthetic organisms they adapt and acclimate to live practically everywhere on our planet. Moreover, despite the fact that photosynthesis is producing practically all our food, the process is not efficient. Less than 1% of the sunlight energy that reaches crops is converted into biomass. Addressing these knowledge gaps will then not only advance basic science, but also the potential to improve crop yields. Historically, the plant cell has not published many papers on the light reactions of photosynthesis and more on the biophysical aspects. And possibly this is due to the perception within the scientific community that this journal does not typically cover this type of topics. But this is not true, and we have organized this focus issue to encourage the submission of the best work in this area, as well as in other areas of photosynthesis, like CO2 uptake, carbon fixation, and improving photosynthesis in crop species. The issue includes two commentaries and a perspective article. The commentary by Ludia et al. Discusses plastid retrograde signaling, and the two articles present a series of mini chapters of vignette written by experts in their areas of research. The commentary by Eckert et al. Addresses 11 open questions in photosynthesis research, while in the perspective edited by me, 12 experts discuss specific aspects of photosynthesis that can be improved. In addition to that, we have 6 in-depth review articles that cover aspects of the major complexes in the light reactions of photosynthesis. Bryant and Gisriel review progresses in phycobilisomes, the antenna complexes of cyanobacteria, rare algae, and cryptophyte. And we want to stress that in plant cell is also interested in photosynthesis in non-plant organisms. Osmaes and all review the structure, biogenesis, and evolution of the thylakoid membranes. And then we have 4 reviews on the main pigment protein complexes of the light reactions. Rollo et al. Review the structure, function, and assembly of photosystem 1. Commenda et al. Cover the biogenesis and maintenance of photosystem 2. Rühle et al. Cover the structure and function of the ATP synthase, and Degen and Johnson review photosynthetic control and the cytochrome B6F complex. We have also 8 actually research articles, and you will hear from the author of 3 of them today. First, Clara Groth-Krego will report on her work on family gene expansion as a genomic driver of CA2 evolution in the genus Tillandsia. Then Ling Zhang will present her work on a new player in cytochrome B559 assembly during early PS2 biogenesis. And David Rollo will report on a new photosystem 1 assembly factors. The other 5 research articles are by Royas et al. That demonstrate the ability of synthetic PPR proteins to activate translation, and they report on the light regulation of PSBA, which is the gene that encodes for the D1 subunit of the reaction center. Xin et al. Present a cryo-EM structure of alternative complex 3. And I take the occasion to stress that the plant cell is very much interested also in structural work. Peltier et al. Quantify the contribution of alternative electron transfer pathways to net photosynthesis in the green alga Chlamydomonas reinhardti. Richer et al. Investigate the regulation of state transition. And finally, Pencil et al. Describe a suppressor screen that they use to investigate functions of photosynthetic related genes. I would like to add that we encourage authors to continue to submit their best work in photosynthesis research to the plant cell, and then the articles that are related to photosynthesis published within 8 to 12 months from this focus issue will still be added to our online collection. With that, I will turn it over to our moderator today, Guy Levine, to introduce the first speaker, and enjoy our webinar.
Thank you, Roberta. So let's start with I will present our first speaker. So our first speaker is Lin Zhang. Lin earned her PhD in 2017 from the University of Chinese Academy of Science. Currently, she's an associate professor in the College of Life Sciences at the Shanghai Normal University. Her research mainly focuses on the regulation of photosynthetic apparatus biogenesis, and she recently identified 2 auxiliary proteins and clarified the regulatory mechanisms involved in the biogenesis of photosystem 2. Today, she will speak to us about the resistance to phytophthora 1 protein, which promotes the cytochrome B559 formation during the early stages of photosystem 2 biogenesis in Arabidopsis. So Lin, the floor is yours. Please share your screen and you can begin.
Okay. Thank you, Guy, for the introduction. I am very happy to discuss our discovery here. Photochrome B559 is a heme-bridged heterodimer composed of 1 alpha and 1 beta subunit. It is located near the D2 subunit. The heme is ligated while 2 is residue, one each from the alpha and beta cytochrome B559 subunits, thereby forming a cross-linked structures. It has been proposed that cytochrome B559 is involved in the cyclic electron transport within PS2. It may accept electrons from QB or PQ and donate them to P680 under photoinhibitory conditions, thereby protecting PS2 from photoinhibition. Another role of cyclone B559 is to initiate the assembly of PS2. Cy-B559 is already present in initial plasts in land plants and initiates the assembly PS2 by forming the D2 cytochrome B559 intermediate, thus forming the cytochrome 559 is the first step of PS2 assembly. Now there are two essential questions on cytochrome 559 that should be clarified. One is the molecular mechanism underlying formation of cytochrome B559. Another is the extraction function of cytochrome B559 in oxygen photosynthesis. In our study, we characterized the RPH1 mutants from Arabidopsis, which was probably shown to be susceptible to pathogen. In accordance with the previous plot, our RPH1 mutants are obviously smaller than WYTAP and other two PS2 mutants, LPA1 and LPA66. While LPA1 is deficient in the biogenesis of D1 subunit, RNA editing of the PSBF transcript is blocked in LPA66. The fluorometric analysis showed that RPH1 are characteristics of photosynthesis mutants with low levels of PS2, such as LPA66 and LPA1. PS2 level was distinctly reduced, and the PSBE is more reduced than other PS2 subunits. Two-dimensional BN page immunoblot analysis showed that free PSBE protein and several assembly intermediates containing PSBE are distinctly reduced in RPH1, suggesting that formation of PSBE containing complex is disrupted in the PSB1 mutant. In addition, we also detected a larger protein band containing PSBE was found in RPH1. Since the amount of PSBE is drastically reduced in PH1 and LPA66 E2 plast, it is likely that RPH1 is primarily involved in the biosynthesis or stabilization of cytochrome B559 during the first step of PS2 biogenesis. RNA-seq analysis and ribosome profiling analysis showed that transcripts level of PSPE and PSBF, and ribosome occupied on the PSPE and PSBF RNAs is not perturbed in RPH1, including the possibility that RPH1 is involved in the gene expression. Protein labeling experiments show that PS2 RC formation is blocked in RPH1, which is likely due to the deficiency of cyclone B509. A pink at 59 nanometer was detected in the spectral analysis, indicating the presence of heme in cytochrome 559. The heme level is more reduced in RPH1 than in LPA66, and only about 60% of PS2 associated with the heme in RPH1, PS2 supercomplex. Thus, RPH1 is likely involved in the proper insertion of heme into the cyclone B559 heterodimer after their transit. Immune blood assays show that RPH1 is an intrinsic silicate protein with 4 precise transmembrane helix, and the N-terminal part of RPH1 is located on the stromal side of silicate membrane. Immunoblood analysis on 2-dimensional BN page show that RPH1 commigrates with PSBE, probably from a complex with cytochrome B559 in vitro. CoIP and YEST2 hybrid and BIFC analysis further show that RPH1 directly interacts with PSBE and PSBF. In addition, RPH1 interacts with D2 in YEST2 hybrid, suggesting RPH1 also participates in the subsequent formation of D2 cyclone B559. Thus, we propose that RPH1 is a detected factor for facilitated non-covalent heme inclusion during cyclone B559 formation in insuloplasty and chloroplast. The next question is, what are the consequences of absence of RPH1 mediated process? As discussed earlier, we found a distinct band in RPH1 which was not detected in Y-type and other PS2 mutants, as well as highlight-treated Y-type and mutants. We purified this band, and immunoblood and mass spectrometry analysis showed that this band contains PSBE and PSBF, indicating that this distinct band presents the covalently linked PSBE-PSBF heterodimer. Furthermore, we found that the level of the PSBE-PSBF heterodimer is reduced when the RPH1 leaves were treated with lincomycin, which blocks the translation of chloroplast protein. This is indicating that PSBE-PSBF heterodimer is likely forming during the biogenesis of cytochrome B559. Thus, in the absence of IPH1 mediate process. Assembled heme molecule may lead to the oxidation of space radius of cyclone B509, lead to the formation of covalently linked PSBE, PSBF heterodimer. Since cyclone B509 is potentially involved in the protein of PS2 under photoinhibitory conditions, in proper incorporation of him into cyclone B559 heterodimer presumably increased. Indeed, all the maculate light conditions, both the FV, FM ratio, and D1 protein level were reduced more in the IPH1, suggesting that an improper cooperation of heme into cytochrome B509 in IPH1 increased photosensitive under photoinhibitory light conditions. We also examined the accumulation of hydrogen peroxide and total IPS levels in the leaves of IPH1 under medial light treatment. The results showed that high level of ROS were detected in the IPH1 than WT, LPA1, and LPA66. Thus, it is likely that an improper incorporation of heme into cytochrome B509 in IPH1 favors ROS production under photoinhibition light conditions. Finally, we investigated the susceptibility of LPA66 and LPA1 mutants to pathogen and found that mutants with low level of PS2 are generally more sensitive to pathogen, although the degrees of susceptibility differs from accident between these mutants. In conclusion, we proposed that IPH1 is a detected factor for facilitating heme encoding during cyclone B559 formation in enteroplasts and chloroplasts without IPH1 mediate process. A covalently linked PSBE PSBF heterodimer is found during the biogenesis of PS2, probably due to the oxidation of specific radici of cyclone B509 by unassembled heme molecule. An improper cooperation of heme into cyclone B509 in IPH1 increased photosensitive and forward ROS production under photoinhibition light conditions. And the last, PS2 mutants are generally more susceptible to pathogen. We genuinely thank all the members contribute to the work and Jane supports this work. Thank you.
Thank you very much, Lynn. If anyone has any questions, please post them in the question and answers box in the bottom of your screen. Okay, so we do have some questions. So first, a question by, I hope I'm pronouncing this right, I'm sorry if I'm not, Dhirendra Kumar. Are there any insights? Why are these mutants susceptible to Phytophthora? Lynn, can you hear us?
Hello.
Yes, did you hear the question?
Excuse me.
Should I repeat the question?
Yes, please.
Dehindra Kumar is asking if there are any insights on why are these mutants susceptible to Phytophthora?
Okay, I'm Liang Weipeng. Guy,
Yeah, can you hear us?
yeah. I'm Liang Weipeng. Yeah, the corresponding author.
Yeah, I know.
Yeah.
That's great to have you aboard. Should I repeat the question?
Yes, I know. Probably the photosynthesis can produce many products and energy, and so this is maybe essential for the susceptible to pathogen. I think this is our idea.
Okay. So we have another question. A quick question. We have time for one more. Is the photosynthetic rate of the RPH1 mutant also reduced?
Sorry.
Is the photosynthetic rate of the RPH mutant also reduced?
Photosynthesis. What's a photosynthesis?
Yeah, that's all right. We don't really have any time to discuss this anymore, so we'll just move to the next speaker. We're out of time. So thank you, Lynn, very much, and thank you for a great talk. And please just stop the screen share, and we will present our next speaker, which is David Rollo. David got his PhD degree at the Max Planck Institute of Molecular Plant Physiology in Potsdam in Germany, where he is now a postdoctoral researcher in the group of Professor Ralf Bock. He investigates how vascular plants assemble their photosynthetic apparatus in chloroplasts. He's particularly focusing on the identification and characterization of photosystem 1 assembly factors. His research involves molecular biology, bioinformatics, and biochemical approaches. And today he's going to speak to us about the co-expressed with photosystem 1 assembly 1 protein and its role in the assembly of photosystem 1 in Arabidopsis. And with that, David, the floor is yours. Good luck.
Thank you for the introduction. Yeah, so indeed, I'm going to talk about this time not photosystem 2, but photosystem 1 assembly, and more particularly about the discovery of a new PS1 assembly factor that we named SEPA1. So, of course, this is a focus on photosynthesis, so you probably already know this, but quickly in this first slide, I will remind you that in Arabidopsis, photosynthesis is sequestrated into the chloroplast organelles. And in the chloroplast, you have thylakoid membranes in which are embedded a few complexes that you can see here, and that mediate the linear electron flow upon light induction. And the last complex is the photosystem 1 that is present here. So before talking about how it is assembled, I will tell you what is photosystem 1. So PS1 is composed of more than 200 ligands, but also many proteins. So you have 14 proteins that make up for the PS1 core. And in addition to this, you have 4 subunits that make the LHC1 that is attached to the PS1. And together, it makes a huge macromolecular complex of around 600 kilodalton. To make this complex more complicated, it has a diverse composition, so the ligands and the proteins. And in addition to this, you have a few subunits shown in green here that are encoded in the plastid, while the other subunits are encoded in the nucleus. So together, this shows that the assembly needs to be very well coordinated to make a functional mature PS1. And how it is thought to happen is in a stepwise manner. So you start quickly with the insertion of the PS1 reaction center, and then you have addition of the stromal subunits. And then with addition of peripheral subunits, you have the formation of a stable assembly intermediate called PS1 star. Then there will be addition of the PSA-F subunits, which will trigger the maturation of PS1 with probably the addition of the LHC1 and the latest few PS1 subunits. And this stepwise assembly cannot take place on its own. It's dependent on a set of specialized proteins, which we call the PS1 assembly factors. And these PS1 assembly factors, they mediate the folding, the incorporation, but also the protection of the PS1 subunit during the assembly. And in the past 3 decades, there are 7 PS1 assembly factors that have been described in plants that you can see here on either sides of PS1. And in spite of this, the assembly process itself is still relatively poorly resolved, especially in comparison to the highly resolved structures that we have of the thylakoid protein complexes. And so this is where we were at the beginning of the study, how to get a better picture of this process. And so the description of these 7 PS1 assembly factors greatly helped to better know how PS1 is assembled. So we wanted to look for new PS1 assembly factors. And this is how we found SIPA1 protein. And now we'll tell you the story of how we found it and why we think that this is a PS1 assembly factor. So similar to the PS1 subunits, you have some PS1 assembly factors that are encoded in the nucleus that are shown in pink here. There are 5 of them. And we know that PS1 assembly factors are mostly co-expressed in young developing leaves, where most of the PS1 needs to be built to make the photosynthetic apparatus. And so we looked for basically genes that are co-expressed with these 5 PS1 assembly factors. And for this, we used the online tool called EnsembleNet. And in this tool, you just give an input of the 5 non-nuclear genes that are coding for the PS1 assembly factor. And the tool is integrating several data, mostly co-expression data. And it will give you an output of genes that are predicted to be functionally related to PS1 assembly. So you get a preliminary list of candidates, and then we narrow down to genes that code for proteins that localize to the plastid and that have uncharacterized function. We ordered a few TAD insertion lines for these candidates, and we got a short list of candidates, among which was, for SIPA1, we got 3 TAD insertion lines here. So that was the first part of the screening to get candidate genes, but now we wanted to have a better view on whether these candidate genes are actually interesting. For this, we looked at the QL parameter by PAM measurements. So the QL parameter is basically the redox state of the PS2 acceptor sites that is present here. And so when you grow the plants and you adapt them to dark, and then you start to apply some lights, there will be linear flow of electron that will happen. And in case of a mutant that have no defect in the photosynthetic apparatus, you will have a nice reoxidation of the PS2 acceptor site, followed by the QL parameter. However, when you have an issue downstream of the PS2 acceptor site, you will have accumulation of electrons, and then the reoxidation will be poorly done at low light intensity. So you will, what we are looking for is the QL phenotype, which is basically this light response curve that we see here. And indeed, for 2 of the mutants, for this SIPA1 genes, we saw this QL phenotype. In red, you have the SIPA1-3, and in yellow, you have the SIPA1-2, which indicated a potential defect in PS1. So that's why we continue to further study this gene. So in addition to this QL phenotype, you can see that the SIPA1-3 and SIPA1-2 have a delayed development, and they have pale green leaves, which is indicative of a photosynthetic defect. So what we did is we isolated thylakoid membranes and performed SDH page and immunoblot. against a SIPA1 antibody that we generated for this study. And indeed, at the expected size of SIPA1 at around 16 kilodalton, you have an absence of a signal in SIPA1-3, and you have a decreased signal in SIPA1-2 as compared with the wild type. And so from this immunoblot, we determined that around half of SIPA1 wild type levels are present in SIPA1-1. There is less than 25% of SIPA1 in SIPA1-2, and SIPA1 is completely absent in SIPA1-3, which is then a knockout. In addition to this, you can see that the QL phenotype that we saw before, the developmental phenotype, both are correlating with the level of SIPA1 in the SIPA1-2 and SIPA1-3 mutant. And in addition to this, by expressing a copy of a SIPA1 flag in the SIPA1-3 background, you can see the nice expression of SIPA1, and it restores here a wild type-like phenotype. So now I just show you that there is a photosynthetic defect in the absence of SIPA1. But however, what makes SIPA1 a PS1 assembly factor? So the assembly factors for PS1 are defined by several features, and here in this talk I will show you 4 key features that are shared by PS1 assembly factors, and I will show you that SIPA1 is sharing these features. So the first feature is that PS1 assembly factors are involved in PS1 accumulation specifically. So what you expect in this mutant is to see only a decrease in PS1, while the other complexes should be only barely affected or as a secondary effect. So for this, we isolated the thylakoid membranes from the mutants and quantified the thylakoid complexes. And as you can see here, you have several complexes embedded in the thylakoid membrane and also the plastocyanin. And in case of photosystem 1, you can see a strong decrease of PS1 in the SIPA1-3 mutant and in the SIPA1-2 mutant. However, this decrease is only mild for the photosystem 2 and the cytochrome B6F. In addition to this, we went for SDH and immunoblot against diagnostic subunits of PS1, but also of LHC1. And you can see the same pattern. All of the PS1 subunits are decreased in the SIPA1-3 mutant. However, the LHC1 subunits are not so much affected, so it's really a defect specific to the PS1 core. In addition to this, for the other complex subunits, there is no major change in the mutant. The second feature is that we expect PS1 assembly factors to not regulate plastigene expression. So PS1 assembly factors are localizing in the chloroplast, and in the chloroplast, as I mentioned before, there are some subunits and assembly factors that are encoded. And so PS1 assembly factors are not active at this level of a transcription or translation, but instead at the post-translational insertion of the subunits. So we did RNA and ribosome profiling, and long story short, there was no significant change in the SIPA1-3 in comparison to the wild type. So based on this, yes, we were thinking, okay, SIPA1 is involved at the post-translational level, so in the assembly of PS1. And so the third key feature was to check that PS1, that SIPA1 is associating with the PS1 assembly intermediate, but that is not a stable component of the mature PS1. So again, we isolated thylakoid membranes, we solubilized with detergent and separated the native complexes by blue native page. And we looked at PSAB, which is a PS1 reaction center accumulation. And what you can see here is that, of course, the mature PS1 is decreased as compared with the wild type, but you also have a decrease in the assembly intermediate PS1 star here, and also potential late intermediate that was not described before. So after this, we wanted to look in the second dimension where SIPA1 is actually co-migrating with. So for this, you have the PSAB here as an example of where PS1 here is. So you can see the mature PS1 here and the PS1 star here. And you can see that in the case of SIPA1, SIPA1 is not migrating, it's not a stable component of the mature PS1. However, it's migrating with several intermediates here, while it is completely absent in the knockout. And it is more visible in the complemented lines here. It's less blurry. You have 3 specific dots, and we annotated these different complexes. So you have the PS1 star and this potential late intermediate here. So if it's SEPA1 is co-migrating with complexes, we expect that it's interacting with PS1 subunits or PS1 assembly factor. So for this, we did a co-IP against the SEPA1 flag and analyzed the data by MS and looked at the enrichment. And indeed, in the enriched proteins, you have PSA3 and PPD1, which are the two other PS1 assembly factors. So we wanted to check if they actually interact directly with SEPA1. So we used the BiFC assay, in which if the two proteins are tagged with a split YFP, and if they interact, basically you have the YFP signal. So we infiltrated transiently Nicotiana benjamina, so for transient expression, isolated protoplast, and looked at the confocal microscope. And indeed, you have a nice YFP reconstitution when SEPA1 and PSA3 are expressed together. However, you don't have this expression when SEPA1 and PPD1 are expressed together. So you have a direct interaction of SEPA1 and PSA3. So in conclusion, we showed you that SEPA1 is a PS1 assembly factor in Arabidopsis. It's associating with PS1 star and probably other PS1 intermediates, including this late assembly intermediate. SEPA1 is cooperating with PSA3 to mediate PS1 assembly on the stromal side. And at the end, we think that it's dissociated from the PS1 assembly intermediate once its function is fulfilled. And so the next step mainly will be to further investigate what is the exact function of SEPA1 and PSA3 together. So I would like to thank all the people who participated to this story. And if you want to look at the full story, you can look at this QR code here. And I thank the organizer for the invitation, and thank you for your attention.
Thank you, David. You can stop. Oh, no, actually, don't stop your screen sharing just yet. Let's if anyone has any question, please post them in the Q&A box. So David, the first question is, what was the light intensity when you compared photosystem 2 and the photosystem 1 protein quantification? If there is high light, there is a possibility that PS2 content may decrease in the SEPA1 mutant.
Yeah, that was just, we just tried the standard light conditions, so around 150 microangstroms. So there was no, we didn't subject the mutants to any challenging highlights or, yeah.
Okay. Another question that we have by Alize Malone. There were 3 bands for SEPA1. Can you say a bit more? Do you think there are truncated products and relevant for function?
So yeah, we think that it's relevant. So I didn't have time to mention this, of course, but we think that they are just a fragment of SEPA1. And what we think is happening is that in the second dimension, you can see the level of SEPA1, the mature protein, and you can still see these fragments, but they're only present in the monomeric fraction. So what we think is that SEPA1, once its function is fulfilled, it's getting cleaved by probably proteases. And then you still find the monomeric fraction, but only you find these fragments, but not associating with complexes anymore, but just in the monomeric fraction. So that's why we think that it's a result of basically the end of a SEPA1 function.
Okay, thank you very much. I think that's good for now. Thank you again for a great, wonderful talk. And now we will move to our last speaker, Clara Groth-Kregel. Clara recently completed her PhD in plant evolutionary genomics at the University of Vienna in Austria. And currently she's a postdoctoral researcher at the group of Ovidiu Paun in Vienna. And Camille, I'm sorry, I'm not sure how to pronounce his name, Camille Roux in Lille in France. Her work is focused on understanding the evolutionary forces that drive adaptive radiations. She will present her PhD thesis research, for which she used genomic data to disentangle the molecular sources of variation underlying adaptive radiation in Tillandsoid bromeliads. Which is a neotropical plant from the Americas. In this work, Laura especially focused on the evolution of CAM photosynthesis as a key innovation trait and driver of diversification in the subgenus of Tillandsia. And today she's going to speak about how CAM evolution is associated with gene family expansion in an explosive bromeliad radiation. So Clara, the floor is yours, and good luck.
Thank you so much.
Please share your screen.
Yeah. Let me see. Can you see my screen now?
Yeah, we can see your screen. You're good.
Oh, okay, perfect. So thank you so much for inviting me today to give a talk. I'm really excited about this. My name is Clara Groot, as Guy already introduced me. My talk is quite a bit different from the two previous talks we've seen. I hope it's interesting for the audience. And it's all about CAM evolution in this bromeliad radiation and how it's associated with gene family expansion. So I want to start by introducing the plants that I worked with during my PhD. This is the subgenus Tillandsia. It's a clade of about 250 species that belong to the Bromeliaceae family, or also known as the pineapple family. And they are considered an adaptive radiation because they diversified very rapidly in the last 2 to 3 million years into hundreds of species. And it's not really well understood how this really can happen at such rapid rates. So that's a very interesting topic for evolutionary biologists in general. And this diversity goes hand in hand with also significant ecological and phenotypic diversity. So in the subgenus Tillandsia, we actually especially are interested in a certain number of key innervation traits. These are traits that have evolved often repeatedly in the group and that are associated with diversification. Examples are the epiphytic lifestyle. So these plants grow on other trees. For example, you also have specific evolved types of trichomes as well, a very large variety of flower color and structure. But what we also have as a listed key innovation trait in this group is the evolution of CAM photosynthesis. So I will actually introduce CAM a little bit later in the talk to catch everybody up on that. But it's a trait that has repeated independently over many plant families in the plant kingdom and is in and of itself a very interesting trait to study for evolutionary biologists because it seems a complex trait, but it has evolved so many different times, making it rather paradoxical. So that's also another reason why we're interested in looking at this trait. The subgenus Tillandsia is not just interesting from a like phenotypic or ecological point of view, but it also like some work has shown that it also seems to have quite dynamic genome evolution. De la Harpe et al. In 2020 looked at rates of gene loss and duplication and found that the subgenus, and especially the Mexican clade, which is the most diversified one, has quite high rates of gene duplication and loss, especially if you compare it to other closely related lineages. And we also have about a threefold variation in genome size throughout the group, and this is without any clear evidence of whole genome duplication. So this is a homoploid radiation generally. And so this made us wonder, is this dynamic genome evolution in any way associated with the ecological diversification that we see in this group? And could we perhaps think that this genome evolution might be underlying this diversification or the evolution of these key innovation traits? And so that's what this project tried to address in some kind of way. We wanted to, first of all, understand better the genomes of these plants. But we also wanted to see whether differences, diversity in the genomes between plants could be linked back, especially to CAM photosynthesis. So to do this, we selected 2 species which represent the phenotypic extremes, let's say, of the photosynthetic metabolisms in the group, starting with Tillandsia fasciculata, which is a strong constitutive CAM plant, and then Tillandsia leiboldiana, which at the time when this project was started, based on carbon isotope ratios, was indicated to be a strong C3 or like a full C3 plant. And the two are quite closely related within the subgenus, so they also underlie a shift in photosynthetic metabolism. So what we did was to get PacBio data, Hi-C data, and Illumina sequencing data, and with that create reference genomes for both species. And here I just want to show you an overview of these two genomes that we generated. They are relatively contiguous, so we were able to obtain about 80% of the reference genome in 25, 26 main scaffolds, which is also the approximate number of haploid chromosomes we are expecting in these plants. And these large scaffolds contain as well, like the majority of orthologous genes that we were able to annotate. Using cytogenetic studies, we also looked at genome size differences between the two species. Tillandsia leiboldiana, the C3 plant, has a slightly larger genome size than Fasciculata. And this is mostly to do with repetitive content accumulation, which is higher in Tillandsia leiboldiana. You can also see that here you see the per scaffold repetitive content in yellow, and you can see that this takes up a really big chunk of the scaffold, whereas in Fasciculata, this is less pronounced. So with these reference genomes then, we wanted to actually compare them and understand how these the genomes of these species are different. So we started by looking at their karyotypes, again with cytogenetic work in this case. And we found that actually there is a significant change in karyotype between the two species, in that Tillandsia leiboldiana has 6 chromosome pairs fewer than Tillandsia fasciculata, and also than the majority of Bromeliaceae, which is quite significant for two species that diversified in the last 2 million years. And as you can see, they also seem larger, these chromosomes, so we started to think that perhaps we have had cases of chromosomal fusion that may explain this difference in karyotype. So that made us really interested in understanding large-scale rearrangements and whether we could define these in our genomes. And so for that, we performed synteny analyses. And for that, we also added to the mix pineapple, which is about 50 million years diverged from these two, and one of the only few other reference genomes available for Bromeliaceae. And as you can see, the synteny, which is based on single copy orthologs and reflects sort of how similar the organization of the genome is between the species, you can see that this is quite well maintained between pineapple and fasciculata. But however, between the two Tillandsias, you see a whole lot of reshuffling, which is, of course, again, quite surprising. We were also able to describe 1 chromosomal fusion, or what seems like a chromosomal fusion, and 2 reciprocal translocations. And then we wanted to ask whether these rearranged scaffolds had a significantly different selection landscape from the non-rearranged ones. And we did that by calculating the DNDS ratios of single copy gene orthologs on all of these scaffolds. And you see here the distribution of DNDS for, in this case, scaffold 14, compared to all the other non-rearranged scaffolds in Tillandsia leiboldiana. And we couldn't really see much of a shift, so it doesn't seem like these rearrangements have really affected the DNDS ratio, so selection on that time scale seems to not be too different. And the reason why we were thinking this might be different is because rearrangements in chromosomes can have an impact on linkage disequilibrium, which then has an impact on selection as well. And based on Marie-Laure de la Harpe's work, we were very interested in gene family evolution as well. And we did see that the rate of gene family evolution was different between the two species, because Fasciculata, the CAM plant, tends to have about 20% more multi-copy gene families than Leiboldiana. And also these are larger in general than in Leiboldiana. But so far, I've not really linked anything back to CAM photosynthesis. I've just been describing how these genomes are similar or different from each other. And for that, I want to explain to you how we address CAM photosynthesis first. And that's first by explaining how it exactly works. So the main difference between a CAM plant and a C3 plant, which is most plants are C3 really, is that CAM has shifted the CO2 absorption to the nighttime compared to C3 plants that do this during the day, and they immediately feed CO2 to the chloroplast and start photosynthesis just in that way. In CAM, on the other hand, CO2 is converted to malate and stored in the vacuole. And then during the day, it will be reconverted to CO2. And that allows the plant to keep its stomata closed, which makes these plants generally also more water use efficient, and is one of the reasons why CAM is often associated with arid and hot environments. And so as you can see, this is mostly actually a shift of temporal activity of enzymes. It's not that it really needs novel enzymes or anything like that. So we were interested in describing the CAM syndrome, let's say, in these plants by looking at differences in temporal expression of genes between the two, and also obtaining metabolomic data over a 24-hour cycle. So that's what we did. We placed our plants in a climate chamber, let them acclimate, and then we sampled them 6 times in a 24-hour cycle. And we obtained metabolomic data from these samples and also RNA-seq data. So the metabolomic data is shown here as a PCA, where we just put all of the compounds that were measured to see whether that already differentiates the 2 species. And you can see that the PCA already has quite a bit of a split between the 2 species, just by looking at all compounds combined. And then we were interested to see how malate was accumulating over time in the 24-hour cycle, because that's a typical feature of a CAM plant. It will accumulate malate as CO2 is converted and stored in the vacuole. And that's exactly what you see. You see a differential accumulation curve for the 2 species, where Tillandsia leibordiana doesn't really seem to significantly accumulate malate over time, whereas fasciculata does show this clearly, showing us that they are for sure different photosynthetic phenotypes. Our differential gene expression analysis found 911 genes differentially expressed between species in a 24-hour cycle, and this included 68 previously described CAM genes. So now we have a subset of candidate genes that are linked to CAM evolution, and we wanted to know if these genes in particular were characterized by some of the genome diversity that or variation that we had already seen in the 2 plants. But for at least the first few that I'm showing here, we couldn't really find a clear signal. So in terms of, for example, TE insertions in or near these genes don't show particularly elevated or lower TE insertions than other genes. We do find that the CAM plant tends to have more often a TE insertion in these CAM genes than Tillandsia leibordiana, but it's only a small signal. We also didn't really find any candidates of the NDS genes, so genes that might have been under selection between the 2 species that were related to CAM. We did find candidates in other pathways. And we also didn't find that they were specifically associated with rearranged scaffolds either. But on the other hand, we did find an association between CAM-related gene families and that they were expanded. More often, they were expanded in Tillandsia fasciculata. So here I'm showing enriched GOTOMs in our differentially expressed genes that are in some kind of way related to CAM photosynthesis. And these bars are showing whether the underlying genes belong to gene families that are more often expanded in fasciculata. That's when the bar are shifting to the right, or when they are more expanded in leibordiana. So meaning more copies in one species or in the other. And you can see that there's quite a few of these functions where there is a shift towards gene family expansion in fasciculata. And on top of that, we also described our genes in terms of what kind of orthogroup they belong to, whether they are in 1-to-1 orthogroups or in expanded orthogroups in the CAM plant, etc. And we found that in our CAM-related DE genes, we have about 3 times more often gene families that are expanded in fasciculata. And we also see an enriched amount of gene families that are multi-copy, but they are equal in size, so their gene duplication is likely to precede the split of the 2 species. So with that, I want to conclude this work. I want to say that we found quite a bit of divergence in terms of the genomes of the 2 Tillandsias, even though they are quite closely related. We found karyotype changes, differential accumulation of TEs, differential gene family evolution, etc. But what we really found linked or associated with CAM photosynthesis was gene family evolution. And on top of that, we are not yet completely finding clear signals that TEs or large-scale rearrangements or cloning sequence evolution are really associated with CAM. So I want to thank everyone involved in this project, the co-authors of this paper, field collaborators, botanical gardens, and you for listening, of course, and I'll be happy to take some questions now.
Thank you very much, Clara. Just to remind everyone, if you have questions, please post them in the questions and answers box. And also to the previous speakers, you can go through the previous questions and you can answer them with writing because we didn't have time to answer all the questions. So first question, Clara, by Maxim. Great talk. Have you looked at animal feeding behaviors of CAM plants? Would animals feed on CAM plants in the evening when starch levels are higher? A bit of a different question. Apologize. It is an interesting question, though.
Yeah, that's super interesting, but I have not looked at this. I don't have knowledge about this actually. It's a cool question. For
Okay,
the Tillandsias, I don't know much about herbivory and Tillandsias, so I'm sorry.
That's all right. And Hugo Lima Melo says he's curious about the next step of the project, if you can describe them a bit. Like if you have a plan, what you're going to do next.
We're currently working on another project that sort of sprung out of this, where we looked at 2 Tillandsia species again, Leiboliana, one of them, and then a different one. And we drought stress them because one thing about CAM is that it's a relatively plastic trait, and it can happen that a plant that performs C3 in normal or like optimal conditions, if it's drought stress, it will activate CAM. And this is something that hasn't been explored a whole lot in Tillandsia because you need to do drought experiments, etc. So we did this, and we found that indeed, actually, Leiboliana can activate CAM. This is also something we already were suspecting from our findings in this work. And then we looked at whether when it was activated CAM, how much overlap was there in the genes that were differentially expressed to the constitutive CAM plant. And we actually found not very much overlap. They seem to be employing different genes even to achieve a similar phenotype. So that's stuff that we're trying to write up now and get published. Yeah.
Well, okay, that's great. Fascinating. And last question, I think we have time for one more. You mentioned the expansion of the ATP synthase family. Can you give more details about what subunits? Are they chloroplastic or are they mitochondrial?
Yeah, I was worried I would get questions like this in this seminar, and I remember looking at this back then, but right now I can't really tell you off the top of my head, but it's in the paper. It's really interesting though. The VATPases seem to have expanded quite a bit, and it also seems like different gene copies were being used differently by the 2 species. So this is definitely stuff to look into more. Yeah.
Okay, thank you very much. It was a great talk and a great study.
Thank you so much.
I think we're done. Thank you to all of our speakers and thank you to all of you that attended. And I don't know, Mary, do you want to say a few words?
Thank you to the editors of the focus issue as well, and thank you particularly to Guy for the great job you did of moderating the questions. We'll be posting this, as I said, to YouTube. In December, we'll be hosting a plant physiology focus issue webinar on the topic of root development, so the other end of the plant. Thank you all very much, and have a good rest of your day. Bye.